Meiosis. Meiosis is the process where a parent cell undergoes two divisions to produce four haploid sex cells, or gametes. Each daughter cell contains half the genetic information of the parent cell and varies in the genetic information they contain. This variation gives rise to species with growing diversity, as each generation of this process is formed through a random combination of both the mother and the father's genes.
Interphase is the phase in which cells normally spend the majority of their time, as it is during this time that cells copy their DNA in preparation for meiosis. During interphase, we can see that the nucleus is bounded by a nuclear envelope and that the cell's chromosomes have duplicated but are in the form of chromatin. Outside the nuclear envelope are two centrosomes, each containing a pair of centrioles. Interphase is divided up into three different stages, starting with the G1 phase. The G1 phase occurs prior to the DNA being synthesized and is where the cell is functioning normally whilst increasing in mass to prepare for cell division. Next is the narrow window of time known as the S phase, during which the cell's DNA is duplicated. Following this is the G2 phase, which is the period after the DNA has been synthesized but before prophase has begun. The mitochondria divide, the cell continues to grow in size, and synthesizes proteins. In prophase 1, chromosomes begin to condense and also start pairing up. Each chromosome aligns with a homolog partner so that a particular gene on one homolog lines up with matching genes on the other. As the nuclear envelope begins to break down, a process known as crossing over then occurs by special proteins breaking the DNA at the same spot on each homolog, while other proteins reconnect the DNA in a crisscross pattern so that the homologs exchange a specific portion of their DNA. These sites of crossing over are called chiasmata. It is this process of crossing over which gives rise to the genetic variation in offspring. Meanwhile, in the cytoplasm, the meiotic spindle, which consists of microtubules and other proteins, forms between the two pairs of centrioles as they migrate to opposite poles of the cell. At the end of prophase, the nucleolus has disappeared and the nuclear envelope has also disappeared, allowing the spindles to enter the nucleus. During metaphase 1, the centrioles have moved to opposite poles of the cell. The pairs of homologous chromosomes arrange themselves under tension on a plane called the metaphase plate, which is halfway between the two poles of the cell. The cell is able to sense this tension and does not progress to the next stage until all chromosomes are properly orientated. Next, spindle fibers from one pole of the cell attach to the centromere of one chromosome of each pair, and spindle fibers from the opposite pole attach to the centromere of the homologous chromosome.
During anaphase 1, the homologous pairs of chromosomes are separated and pulled apart by the spindle fibers attached to the centromeres of each chromosome. The pairs of chromosomes move towards opposite poles. It should be noted that in meiosis 1, sister chromatids, called dyads, remain joined together after metaphase. This is a key difference between meiosis and mitosis, where sister chromatids would normally separate.
In telophase 1, the spindle fibers allow for the complete migration of the homologs at opposite poles. There are now haploid sets at each pole, and new nuclear membranes form around each chromosome set. Following this, cytokinesis occurs. This involves the formation of a cleavage furrow where the cell splits into two. In each of the daughter cells, there is a nucleus with a haploid set of replicated chromosomes inside. Following this phase, the cell proceeds into meiosis 2. Prior to the beginning of meiosis 2, no further chromosome replication occurs. During prophase 2, the centrioles duplicate and two pairs of centrioles separate into two centrosomes. Within this phase, you can see the nuclear envelope disintegrate and spindle fibers reform.
In metaphase 2, the chromosomes align along the metaphase plate, similar to the process in metaphase 1. However, rather than the homologous chromosome pairs present in metaphase 1, only two single chromosomes are present in each cell. The spindle fibers attach to the kinetochores of the sister chromatids in preparation for the division at the centromere. During anaphase, sister chromatids are pulled apart at the centromere by the spindle fibers and move towards opposite poles of the cell. This differs from anaphase 1, where the homologous pairs of chromosomes separate, while sister chromatids remain attached. In telophase 2, a nuclear membrane forms around the chromatids gathered at each end of the cell. Cytokinesis divides the two cells to produce four haploid cells, each containing 23 chromosomes, which is half the genetic information of the parent cell. The gametes produced range in genetic composition due to the process of crossing over in meiosis 1, and the random arrangement of chromosomes, creating diversity in offsprings.
Come on. Let's talk about sex, baby. Let's talk about you and me. Let's talk about all the good things and the bad things that make me. Let's talk about sex. Let's talk about sex. Let's talk about sex.
Yum, mitotic spindle.
Oh, get some chromosomes.
Centrioles. I just fuck chromosomes.
Tastes like meiosis.